Okay, this is Chapter 3, Transmission of Genes from Generation to Generation. So it'll be a brief overview of the PowerPoint, and this is in no way is intended to make up for reading the textbook, but I just wanted to go over the PowerPoint slides with you quickly and give you some commentary on some of the sections for this chapter. Okay, here we go. Heredity. How are traits inherited? Well, first of all, why do we begin by examining inheritance and discussing Gregor Mendel and his pea plants? You may or may not know Gregor Mendel or remember him from biology, but he was the Austrian monk that did the pollination, cross pollination with the pea plants and discovered a lot of things about heredity. So, before Mendel really experimented with the inheritance of traits in garden peas, there is really no clear understanding of how traits were inherited and passed from one generation to the next. So Mendel used experimental genetics to uncover the fundamental principles of inheritance. Mendel's experiments established the foundation for the science of genetics and defined the ideal properties of an experimental organism. These were that it should have a number of different traits that can be studied, that the plant should be self-fertilizing and have a flower that reduces accidental pollination, offspring of the self-fertilized plant should be fully fertile so more crosses can be made, and that you have a large sample size. He used more than 28,000 plants in his experiments. There's a little picture, modern day, of his garden or where he did some of his gardening at the monastery in Austria. Now, the traits that Mendel used or that he looked for when he did his studies were with peas were shape, color, pea coat color, the shape and color of the pods, the position of the flowers, and the length of the stems. And you can see that he was able to ultimately identify some dominant and recessive traits. The dominant traits, for instance, for peas, for shape, were smooth. For color, it was yellow, and for pea coat color, it was gray, whereas the recessive for the peas shape were wrinkled. For color, it was green, and for pea coat color, it was white, and so on and so forth as you go down the table. Now, Mendel's initial crosses studied the inheritance of a single trait, such as shape or seed color. And when plants with smooth peas were crossed with plants that having a wrinkled skin, the offspring all produced smooth peas. Now, when the plants from these smooth seeds were crossed with each other, then you get a different result. They weren't all smooth. They yielded 7,324 peas in total, of which 5,474 were smooth and 1,850 were wrinkled, so they weren't all smooth. He utilized some terminology. He called the first generation the P1 or the parental generation, and then the offspring of the parental generation was considered the first generation or the first filial generation, and then he used F2 or second filial generation for the grandchildren of the parental generation, if you would. So in this experiment, in the first generation, the parental generation, parental generation, he used smooth and wrinkled. He crossed them. And then the first generation, the F1, he got all smooth. And then the F2, 5,474 were smooth and 1,850 were wrinkled. As you can see here, it shows the result of his cross and the F2 generation. Some of the different traits that he looked at. Again, there's the 5,474 smooth, 1,850 wrinkled, etc. And obviously there's some different characteristics there. So, but we're going to focus on this seed shape right now. Okay, so again, in the smooth and wrinkled cross in the parental generation, the first generation produced smooth, which he self-fertilized those. And then in the second generation, you got 5,474 smooth, 1,850 wrinkled. So the total for the second generation was 7,324. Now these numbers are actually kind of important. Not that you need to have them memorized necessarily, but they're important because later on they'll show statistically that you get a 3-to-1 ratio. Now, only one of the parental traits was expressed in the F1 plants. Genetic factors could be hidden, they could be unexpressed, and despite identical appearance, the P1 and F1 plants must have different genetic factors. So the F1 plants must carry something hidden, some hidden genetic factors. The trait not expressed in the F1 plants reappeared in 25% of the F2 plants. So that meant that the F1 plants must carry two factors, one from each parent, and that the traits were not blended as they passed through the parental generations. In these crosses, there were two inherited elements of factors that were responsible for the trait. These factors are now referred to as genes. In the F1 generation, these factors or genes were present, but they were not expressed. And so the terminology dominant trait and recessive trait comes as a result of this. The dominant trait is the trait that's expressed in the F1 condition, and that's considered heterozygous because both of the alleles, which you'll learn about here in a moment, were not identical. They're different, so they're hetero. And the recessive trait, the trait repressed in the F1 but re-expressed in some members of the F2 generation. You'll see more on that in a second. Now, a couple other terms that you need to know. Phenotype are the observable properties of an organism, and the genotype is the specific genetic constitution of an organism. Now, keep in mind that some traits can appear in offspring even when the parents don't have the trait. So here you can see in the parental generation, now we're going to take a look here at the gametes or the sex cells. So if you look at the genotype, the phenotype for instance is smooth, and the genotype, if they're homozygous dominant, they're going to be capital S, capital S. Now, only one of these alleles will get produced in the offspring. So you'll see here the S and S get separated next to the word gamete. So there's a big S and one green smooth P and a big S in the other green smooth P. And then meiosis is going to occur between the smooth and the wrinkle. And the wrinkle is represented by the little s's because it's homozygous recessive, so little s and little s. And now the only opportunity you can get when you cross these is a big S from the smooth and a little s from the wrinkled. And so the genotype in the first generation is going to be big S, little s, and because the big S is dominant, the phenotype is going to look smooth. But even though if you look at this F1 and compare it to the P1, you can see that in the F1 you have big S, little s, and in the P1 for the smooth, it's big S, big S. Now, these are different. Again, the P1 for smooth is big S, meaning it's homozygous. Homo for alike, and the big S's are identical. And in the F1 generation, you have big S, little s. It still looks smooth in the phenotype, but the genotype's different because you have a big S, a little s, and this is called heterozygous, hetero meaning different. Now, pairs of factors or genes separate from each other during gamete formation and exhibit dominant or recessive relationships. Genes are the fundamental units of heredity, and segregation means that members of a gene pair separate from each other during gamete formation. For instance, if you cross in the F1 here on the left side, a heterozygous smooth with a heterozygous smooth, so a big S, little s, crossed with a big S, little s, you can only get from each parent a big S or a little s. And so gamete formation by F1 parents means the big S, little s will yield a big S gamete and a little s gamete. For the male and for the female, a big S gamete and a little s gamete. And so when you cross them, you could get a variety of options, like it shows here in the F2 generation. The genotype could end up being big S, big S, little s, or little s, little s. And if you notice, big S, and big S, little s, phenotypically all look smooth, even though genotypically they're different. And the little s, the genotype and phenotype will be the same. They will be wrinkled because the only way to have that wrinkled characteristic or phenotype is if it's a recessive, and that means that they has to be homozygous. They have to have little s and little s in the F2. Now, alleles are possible alternative forms of a gene. They're usually distinguished by the phenotypic effects. Homozygous means having identical alleles for one or more genes, and heterozygous means having two different alleles for one or more genes. So, like you saw earlier here, if you look here, and try to do this with my pen here, if you look over here, you have big S, little s for the male, and big S, little s from the female. So they both look smooth because they have this big S, and that's dominant over the little s. So you're going to see the big S, you're not going to see the little s. So you're going to look smooth phenotypically. Now, the possibilities for each of these, if I take this male and I bring it over here, and I take the female and I bring it over here, the possibilities are I could have a big S, I could have a little s, and for the female I could have a big S and I could have a little s. Now, what we're going to do is we're going to take these, and again, this is the male, so we're going to take these and we're going to put them right here. So you have big S and little s. And this is called a Punnett square, by the way. And then for the female, you have, again, big S, little s. And we're going to put those right here. So we have big S, little s. Now, if we look at these and we cross them, the only possibilities are we could get big S, big S, big S, little s, big S, little s, and then little s, little s. So I'm trying to draw them bigger and smaller so you can see. So this is called the Punnett square. And then what you can see is this big S as we have here is going to become smooth. Now in this case it's smooth, but it's going to be homozygous smooth because you have alike letters, or both capital S's. Now over here in this one you're going to have, again, smooth phenotypically, but they're not going to be the same. So those are going to be called heterozygous for the trait. So big S, little s. They don't have two big S's or two big little, or it's going to be two little s's. That means they're heterozygous because they're different. So again, the genotype is going to be different. It's big S, little s from this one here where it's big S, big S. So this is homo, meaning alike, and this is hetero, meaning different, even though phenotypically they're smooth in either case, right? Now, oops, sorry about that. Now, also here, if we look down here, we have big S, little s, which is going to be a similar situation to up here. Did that again, sorry about that. So genotypically, they're going to be heterozygous. They're going to be different, but they're still phenotypically going to look smooth. So if you notice, you have three smooth here. You have three smooth, and we'll talk about that again in a second. Let me finish this up. You have little s when you come down here, so that's going to be homo, but it's going to be homo recessive because it's the little s's. And now because of that, because you have two little s's, only when you have two, when it's a recessive trait, you have to have two lowercase alleles in order for the recessive phenotypic trait to be expressed, in this case wrinkled. So here this was hetero as well, right? Hetero. So you have, if you look here, you've got 1, 2, 3 out of the 4 are smooth, and then 1 out of the 4 is wrinkled. And you had 1 big S, you had 1, 2 big S, little s's, and then you had 1 little s, little s. Now this is homozygous recessive. Now this one here is homozygous dominant. And you can tell whether they're dominant or recessive based on whether the letters are capitalized or not. If it's capitalized, it means it's dominant. If it's not capitalized, lowercase, it's recessive. In this case here now, because we have a big S and a little s, this is called heterozygous. Now in this case, the trait, the phenotypic trait, is going to be smooth, just as if it were, keep doing that, sorry, as if it were homozygous, because as long as you have one big S, that's the trait that's going to be expressed because it's dominant over the recessive trait. To have a recessive trait be phenotypically expressed, in this case wrinkled, you have to have two lowercase. So I hope that makes some sense and helps out. Okay, now next concept is Occam's razor, and what that simply means is, well, as they say here, what William of Ockham said. I think he was a monk in England back in the turn of the Dark Ages into the early Middle Ages, I guess, 1300, 1349. But he said entities must not be multiplied without necessity. In other words, Occam's razor, if you've heard of it before, is generally termed as the simplest explanation is usually the most correct or likely to be the correct answer. It's also known as the principle of parsimony. And it basically goes like this. When constructing an argument, use the smallest number of steps or the simplest assumptions. And the simplest assumption is usually the easiest to prove or disprove scientifically. So make sure you're familiar with Occam's razor and that concept. Okay, more pea plants, multiple traits, the principle of independent assortment. So Mendel's later experiments show that the members of one gene pair separate or segregate independently of other gene pairs. So now what we're looking at here in this section is when you do a cross, we're looking at not just smooth and wrinkled, but we're combining traits. Smooth and wrinkled, but also yellow and green. So here we have smooth yellow and wrinkled green. And the parental generation. The first generation of offspring, you have all smooth yellow. But when you cross the smooth yellow with a smooth yellow, you get something entirely different in the second filial generation when you're looking at two traits. You get 9/16 smooth yellow, 3/16 smooth green, 3/16 wrinkled yellow, and 1/16 wrinkled green. So what this shows is that all of the offspring are independently assorted, that yellow and green can work independently of each other as well as smooth and wrinkled. So this is called the law of independent assortment, which is the random distribution of alleles into gametes during meiosis. This leads to the formation of all possible combinations of gametes with equal probability and a cross between two individuals. So here's a very small picture of the Punnett square looking at a dihybrid cross, but let's take a let's take a better look here at the picture. Let's take a look at this one first, and we'll go to the next slide. So it's kind of split up between two slides. But here we go, the parental cross. Let's say we have a smooth yellow and a wrinkled green on the left. If you notice here, from the smooth yellow, you have capital S and capital Y. So the only thing you can get, these are identical, so the only option you can get is capital S. And from here, between the two Y's, if I'm only going to get one of these out, the only thing possible is a capital Y. Now here, if you look, again, you have a lowercase s and a lowercase y. That's all you can get out of this. So now when we cross these, you're going to get big S, little s, right here, and then big Y, little y, okay? And that's going to look, because the big S, which is smooth, is dominant. It's going to be smooth. And the big Y here for yellow is dominant over green. It's going to look like smooth yellow. Okay, so over here you have another one. We have a wrinkled yellow and a smooth green. Now from here you can only get a little s, and it's yellow, so you get a big Y. And over here you get a big S, and for this allele you get a little lowercase y. So you have smooth green, wrinkled yellow, and when you cross them, you can only get big S, little s, big Y, little y. So if you notice that, look at that. You have big S, big Y, little y from this side, and over here you get big S, big Y, little y. But yet the parents looked entirely different. They were smooth yellow and wrinkled green, and then you had the opposites, wrinkled yellow and smooth green. But when you look at the possibilities for the cross of the first generation, they're identical. So now let's take a look at what happens. So when you cross these, again, we take one of the parents, the male, we put them along the top, put the female along the side. So what can I get out of here? I can get four possibilities. If I look at this, it's kind of like doing your FIFA or FIFO in math when you do algebra. You have first, inner, outer, last. So here we go. S, big Y. Okay. Big S, little y. Little s, big Y. And then little s, little y. Those are the four possibilities I can get out of this. And the same thing with the other parent, right? So you go down this side, and you're going to get big S, big S, big Y, big S, little y, little s, big Y, little s, little y. Okay, now we're going to go ahead and cross these. So you get big S, big S, big Y, big Y. So you get that here, and that's going to look phenotypically like smooth yellow. Here you have big S, big S, big Y, little y, so you get smooth, again, yellow. But again, genotypically different, phenotypically the same as the predecessor. So here, big S, big Y, smooth and yellow again. Again, a different genotype, but same phenotype. Big S, big Y, little y. Again, um, another phenotype. Excuse me, same phenotype, but another genotype. If you look at these here, all four of them are genotypically different, but they all look like smooth yellow. And so again, you go through and you just do the boxes. So big S right here, big Y, little y, okay? And you get smooth yellow. Here you get big S, big little y. So here you get big S, so it's going to be smooth, but you have the recessive trait for color. So recessive leads to a green coloring. So it's smooth, but it's going to be green. Here you get big S, little s, and then you get little y and big Y. So you get smooth yellow again. And then here you get big S, little s, and you get little y. So again, smooth, green, but again, a different phenotype, same here between these two, but genotypes are different if you notice. One's homozygous here for the smooth trait, and this one's heterozygous for the smooth trait, and so on and so forth. You go through and you just cross these, and ultimately you get these values when you look at them and you do it all out. You get 9/16 of them are smooth yellow, 3/16 of them are smooth green, 3/16 of them are wrinkled yellow, and 1/16 are wrinkled green. So you get this 9 to 3 to 3 to 1 ratio. And that's what you generally get for the F2 generation of what's called a dihybrid cross, because we're looking at two traits, so it's di. Sorry about that. Dihybrid cross. Okay, and you get this 9-3-3-1. If you remember from the other type of cross that we did previously, you had a 3-to-1 ratio, right, at the F2 generation. And then here you have a 9-3-3-1 at an F2 generation. Another way of looking at those same values. Okay, what do we do with these? How do we get some information out of this stuff if we were to do these types of, collect this type of data and take a look at it statistically? What could we identify out of this? Well, there's a simple test utilized pretty heavily in biology called the chi-square test. And what this is it is a statistical test to determine whether the observed distribution of individuals in phenotypic categories, in other words, the way they look, phenotypic categories, the way they look, is as predicted, or if that just randomly happens. So, is what we are seeing the result of chance, or is what we are seeing the result of some actual instance occurring that's causing this? So the chi-square, x squared, that's the chi-square right there. X is chi in Greek, so it's chi squared is equal to the sum of the, I think it's the difference squared divided by the expected result. Now, you don't necessarily need to know how to do this. Just know that there is a chi-square test that's available to take a look at. Is something occurring randomly, or is it actually occurring as a result of some instance, some event that's causing it to happen? So accepted probability will also depend on the number of phenotypic classes. So to get the number of phenotypic classes, what you do, it's called the degrees of freedom, or DF. It's going to be the number of different classes you have minus 1. So let's say if you looked at 8 different classes, or 8 different phenotypes, or 8 different ways something looked, it would be 8 minus 1, so the degrees of freedom you would use is 7 when you go to apply it on the chart. So if you calculate your chi-squared value, you would utilize that, and you would also utilize your degrees of freedom to go to a chi-square table and look at the data to see if it's a probable chance or if it's just a random event. Now, you take a look at, here's some chi-square analysis of some of the data that Mendel did from some of the earlier experiments before. So you can see here, it's quite simply to get D, the value D, which is right here. Okay, here's your D, and notice that they square it. They square it so that way, no matter what number it is, whether it's positive or negative, it's going to be a positive value. So a positive value squared is still going to be a positive value, but if it's a negative value, you don't want to necessarily have a negative value there, so you square it and it becomes positive. So when we go here and we take a look at this, how to get D, and here's D again, right over here, it's going to be the observed minus the expected. So if you look at the observed, Mendel observed 315 yellow smooth. Now, based on the probability of these ratios, 9331, and the total number of plants that he worked with, 313 should have been smooth yellow. He actually got 315. So if you look at this, observed 315 minus the expected 313 is going to be equal to plus 2. Over here, smooth green, he observed 108. He expected to have 104, so 108 minus 104 is plus 4. And then wrinkled would be 101. Expected would be 104, so 101 minus 104, negative 3. But that's why we square it, because that's D. But when we make it D squared, it would be negative 3 squared, which would be positive 9, right? So you get a positive value. So when you go and you stick it back in here, that would be 9 divided by the expected value. In that case, sorry, it would be expected would be 104, so 9 divided by 104. And then you would add all of them up and get a value, and again, calculate your degrees of freedom. Here, your degrees of freedom, there would be 1, 2, 3, 4 options, so it would be 4 minus 1 or 3. Add the sum of all your D squared divided by your expected, and go on to the chi-square table, plug it in, and then to see what level of I can't say the word statistical probability you are at. Okay, so moving on. Okay, so let me see here. There we go. Okay, so um, Mendel's principles of segregation and principles of independent assortment are fundamental to our understanding of the science of heredity, also known as genetics. So keep in mind, we can identify genetic traits because they have a predictable pattern of inheritance that have been worked out by Gregor Mendel. Now, meiosis explains Mendel's results. Genes are on chromosomes. So gene pairs or alleles are located on chromosome pairs. The position occupied by a gene on a chromosome is referred to as its locus. The behavior of chromosomes in meiosis causes segregation and independent assortment of the alleles. So here you can see genes, chromosomes, and meiosis, table 3.2. In genes, they occur in pairs. They're known as alleles. On chromosomes, they occur in pairs, but they're called homologs. The next group, genes, members of a gene pair separate from each other during meiosis. And for chromosomes, members of a homologous pair separate from each other during meiosis. And then the third grouping, members of one gene pair independently assort from other gene pairs during meiosis. And on chromosomes, members of one chromosome pair independently assort from other chromosome pairs during meiosis. What this means is something like this. So if you have in meiosis 1, let's take a look here at the left side. Sorry, touched the tablet too early. So if you look here, you've got a chromosome here, chromosome A, big A, dominant A, little recessive A, and you have B and its version here. So let's say here you get A and B get pulled out here. So A and B. So you get this one and this one. Now, you're going to get 1/2 of these in meiosis, because in meiosis you're going to cut the values in half. So you get a big A, and you get a little b, and then the other one big A, and the other one little b. And then on this side here you get the little a, get a little closer, little a, little b. So you get little a, little b. So you get little a, little b here, and you get little a, little b here. So these are the gametes, the sex cells. They're independently assorted. In other words, they don't get grouped together. Just because they, like, you don't put the B's together, for instance, you don't put the A's together. They break apart into halves, and so you get one half in each of the gametes. Okay, same thing over here with meiosis. Now here we get a different order. You got big A and little b rather than big A, big B. And over here, little a, big B rather than little a, little b. So now when we get these, we get A and B, little b. So they break out to A, little b, big A, little b. And on this side here, you get little a, big B. So you get little a, big B, little a, big B. And so you get various options. Notice they're all different sets here of gametes because they independently assort. They don't always go together. Now, segregation and independent assortment occur with human traits. Inheritance of certain human traits is predictable based on the Mendelian inheritance of patterns. Here, segregation of albinism. On this slide, we're taking a look at, we've got two parents. Let's look up here. They are heterozygous for albinism. We're going to say to become albino, you would have to be little a, little a. It's a recessive trait. It's homozygous recessive because you would have two little a's. So what you get here from the male and from the female, you get, what can I get from this parent? Well, it's big A, little a. Mom, you can get big A, little a, right? So big A, little a, big A, little a when they independently assort. So when I go down here, I put the male up top so I could get big A, little a, and then I got big A, little a for the female on the left side, the mom. And now we put big A together. That would be normal. As long as they have at least one capital A, it's going to be a normal individual, although heterozygous. Now this is a homozygous normal. But let's take a look here. Big A, little a is going to give you, again, a normal individual, but heterozygous. And then big A, little a here. You have a heterozygous normal individual. And you have little a here, and you have a homozygous individual. And we said little a is going to mean you're going to have albinism. In this case here, that's the albino. So if you look at this, 3/4 of the offspring will be normal. Now they may have different genotypes. It could be big A, or big A, little a, but they're going to look normal. In the case here of the other child, that 1/4, you have a, and that means they will have albinism. Now, this can also, apart from obvious features of albinism, are like the light skin, light hair color, reddish eyes, so there's a pigment deficiency, but you could also have hearing deficiencies associated with it too. So if you want to take a look at these from a dihybrid perspective, you could look at the two different types of genes or two different alleles, and do a dihybrid cross like here, and you can see how many of them will be normal with normal hearing, how many will have normal pigment but might be deaf, how many of them will be albino but have perfectly normal hearing, and how many will be albino and deaf as well. Now, pedigree charts are used in humans to trace generations, to look at relationships in a family, and to study heredity. They look like these boxes and balls, as you have probably seen in biology classes in the past. And so a pedigree is a diagram listing the members and ancestral relationships in a family. It's used in the study of human heredity. A couple quick things to note here. Generally what you see is if you're a box, that's a male. If you're a circle, that's a female. And the line between the two, meaning that they had some sort of relationship in which provided offspring as a result of mating, and that this line here shows their children. So this male and this female produce these 4 children, and the 4 children are 2 females and 2 males. So 50-50 male, 50-50 female as far as a sex split. And then usually if they're darkened, if it's a blackened box or a blackened oval, or a circle, that just means that they have a particular trait. Okay, so here you would see you have a daughter without the trait, a daughter with a trait. male without a trait, um, and then a male with a particular trait or disease or whatever you happen to be looking at. So, constructing a pedigree, you use standard form and symbols. There's a set program for how to do all of this type of work. You use family history to show how a trait's inherited and estimate the risk for family members, and it helps genetic counselors in providing genetic counseling for those at risk of having children with genetic disorders. So here's some of the diagrams and terminology and pictograms that are utilized in doing pedigree charts. Um, don't get too worked up over these. If you know male, female, and whether they're affected or not, and then who the proband is. Usually, the proband is the first person that actually goes to the doctor that reports a trait. They're the first identified person, and you can kind of work off of them to help identify other individuals in a pedigree chart. But that's probably good if you know male, female, and affected versus non-affected and proband. That should probably work. Okay, each generation is identified by a Roman numeral. So here, for instance, the first generation, the parental generation, if we were using Mendelian terms, P1, right? So that would be Roman numeral 1, and then the first filial generation, the F1 generation, would be Roman numeral 2, and then each individual within the generation is identified by Arabic numerals. So here you've got individual 1, individual 2, and then their children would be child 1, child 2, child 3. Usually they will list these in order of birth. So, for instance, daughter 1 was born between son number 2 or child number 2 here, and then there's a daughter that was born last. Now here you can see that the females were not affected, but the male was affected by something, which means the only way for this person to get the recessive trait, let's say it was, use something that's a little different, like y, lowercase y, right? Means you can't have parents that were big Y, big Y, big Y, big Y, because there's no way the children could get this, right? Because each parent would have to contribute something to that child. That means when you work these backwards, that this parent was probably big Y, little y, and that this parent here was big Y, little y. And that way, for this child to have the trait, they would get one Y from mom and one Y from dad, and that's the only way that this would happen. Now, these children here, you don't know. They could have gotten the big Y from dad and the big Y from mom, so they could be big Y, or big Y, little y. Same thing over here. You just don't have enough information to tell. Sorry, I'm drawing so sloppy on this. Try not to hit the cursor. All right. Oh, and the last thing here, the first affected family member who seeks medical attention for a genetic disorder is referred to as the proband. Usually you write a P and you draw an arrow to which box it is. In this case, that's the individual right there. That's the proband, number 2. Okay. All right. So here they're saying, follow a trait through a pedigree. You can kind of work back. So you can see here if you've got like affected individuals, that means this here, the situation between these two mating individuals, um, to have affected child, if it's a recessive trait, right, they're going to have each be carriers for that recessive trait, meaning they would, in the case I used previously, have a lowercase y, or something like that, right? So we know that is going to happen, and so on and so forth. So you can kind of work these things backwards and usually figure out a good deal of the information, especially the more you can collect in a family history from a patient, the better off you are in figuring these out. So keep in mind, pedigrees are constructed to follow the inheritance pattern of human traits. And variations from Mendel. There's a couple last things to talk about here in this chapter. Alleles can interact in ways other than dominant recessive. They could be incomplete dominant, codominant, or have multiple alleles. And different genes can interact in different ways via a um, uh, state known as epistasis. I guess that's probably the best way to put it. It's not a term that's used loosely. Basically, epistasis, the easiest way to think about it is if you have twins that are identical, like you have two Swiss sisters, um, they could look very different from each other, even though they are twins, um, identical twins, even that there's still enough characteristics that are different between the two. It's a result of certain genes being turned on in one child and perhaps not the other, or at least not at at a certain point in development, and so it causes differences in characteristics. They don't look exactly the same. So that's probably the simplest way to explain epistasis. It's looking at other means of genetic control that can turn genes on and off at different stages of embryonic development, or even throughout life. Okay, let's take a look at what we're talking about up here in the first bullet: alleles, um, interacting other ways apart from dominant recessive. Let's look at incomplete dominance first. So incomplete dominance, here you have the expression of a phenotype that is intermediate to those of both parents. An example is the inheritance of flower color in snapdragons. For instance, if you have a parent that's dominant for red, homozygous dominant, and you cross it with a female that's, let's say, homozygous for white, then the offspring is going to get a codominant trait from, or excuse me, a trait, a gene from each parent that is going to be, how to put it, intermediate, I guess. So here in this case, let's see, you've got the father, right? You can only get R1 from this individual, right? Because that's all that they can provide. Here you can only get R2. So when you cross them, you can get R1 from one father, R2 from the mother. So the child's going to be R1, R2 in this case, right? Just like they're showing here. And because it's incomplete dominance, right? So one trait doesn't necessarily dominate over the other. You get a blending of the colors. So if one's red and one's white and one doesn't overtake the other, you end up getting this blend. So red and white makes pink. And so the offspring there, this is the R1, R2, is actually a pink color, right? So it's incomplete dominant. Codominance, you have a full phenotypic expression of both members of a heterozygous pair. An example is the inheritance of the MN blood group in humans. For instance, the genotype, if you have LMLM, you're going to have blood type M because that's all you can get from it. If you have LMLN, you can only get blood type MN. And if you have LNLN, you can only get blood type N. So this is an effect of codominance. Even though you have M and N, they are both equally dominant. So both get expressed. This isn't a blending. This is like you get M and you get N, right? This is not like M's dominant over N, so N doesn't show up like normal. They're equally expressed. So this is not red and white making pink. This is actually, this person's like red and white. Kind of like a mosaic. Okay, multiple alleles. Genes that can have more than two alleles. We've been only talking about two traits so far. But what happens if there's like more than two alleles for a particular trait, like blood type? In this case, the alleles are codominant. So if we look at A, B, and O blood types, right? So there's three different possibilities. You can be A, B, or O, different alleles. So for instance, you could be, to be type A blood, you could have A or A and the recessive I, right? So this is right here. You have A expressed, or you have A expressed but in fewer number, right? Because the other one's the I, which is the recessive trait. Same thing for the B's. You could be homozygous dominant for B or heterozygous for B, in which case you get this. And you could be codominant here, where both, excuse me, A and B are expressed, like this individual. Sorry, this individual right here. Sorry. Okay, they're both expressed, so you have both A's and B's on it. Or you could get little I, in which case you have type O. You don't have A or B expressed, so you have a totally different type. You have type O. Okay, you have the two recessive traits, so A or B not expressed, you have type O. Okay, finally, epistasis, a form of gene interaction in which one gene will mask or prevent expression of another gene. It could turn something on, turn something off. Somehow, expression of a gene could be masked by another. An example is the Bombay blood type in humans. The Bombay gene, which is unrelated to the ABO blood type gene, when mutated, can block expression of blood types A and B, and so therefore they look different than what you would normally expect. And that's the end of chapter 3. Thank you, and there'll be more shortly coming.